Hello and welcome to another Piper Pod. Today we're going to continue along the anatomy physiology and we're going to have a two-part series looking at muscle physiology. The first part is going to cover an overview on muscles, focusing mostly on the skeletal muscle. So go through the non-science, really, the non-microscience anyway, of muscle physiology. The second part will be focusing purely on the contraction itself. So the muscles structure of our body, the skeletal muscles constitute about half of our body weight, obviously different for some people, but the functions itself are obviously involved with body movement and the maintenance of posture. So body movements are mostly your voluntary movements throughout your body, whereas your maintenance of posture is, whilst controlled by the skeletal muscles, it's sort of taken care of with the help of proprioception fibers. From there we have a assistance with organs, mostly respirations, but then also the return of blood through venous return with smooth muscles. And something that most people don't think about, the actual muscle systems are responsible for our communication. Like how we talk to one another is all got to do with muscle control. And most of the heat produced in our body is actually from muscles itself. Not as a byproduct of energy production that you learnt last time, but actually muscles itself generate the most heat in our body. So things that are quite unique about muscle structures, when we look at the cellular structure of muscles, there's four relatively unique properties that we can look at. So first of all, obviously they can contract. And with that contraction, they have the ability to shorten, but they have the ability to shorten with the generation of force. Okay, so they're not just going through a motion of going from something larger to smaller. As a result, they have an output of force. Next one is they're excitable. Okay, so they actually could be considered an extension of your neural tissue. Okay, so they can respond to the same sort of stimulus and have a similar pathway of travel within them that you'll find in the nervous system. Okay, next one is they're extensible. So the muscle can stretch well beyond its capacity. That helps it in the ability to elongate under certain stimulus without essentially being broken. Okay, so it's quite useful. And this is controlled by other nerves called the Golgis. And the Golgis themselves will tell us when the muscle is stretched to capacity, and it will also go through an initial contraction. Okay, that will be the stimulus itself. That's what happens when we stretch. Okay, and the last unique ability is they're elastic. Okay, so after they're stretched or after they're shortened, after they go through contraction, they have the ability to recoil back into their original shape by themselves. Okay, we don't have to mold them back into their original shape. Okay, so that's what is unique about muscles themselves. Now there's three different types of muscles in the body. We have the cardiac, the smooth, and the skeletal. I'm going to mention the cardiac and smooth, but today we're going to purely focus on skeletal muscles themselves. Okay, so cardiac muscles we've spoken a bit about in the past. They are striated muscles, just like skeletal, but branched a little bit different. Okay, so they're connected by intercalated disc, and they only have 1 to 2 nuclei per cell, which is, and again, different to skeletal muscles that we'll find. Good thing about the heart muscles is obviously they're involuntary, they're controlled by a pacemaker, and they're what's known as a medium speed of contraction. Okay, so whilst the contraction can speed up, it's not as fast as skeletal muscles, if you will. So if you imagine blinking, okay, that's skeletal muscle control, and that is extremely quick in its reaction. The other extremely positive thing about the cardiac muscle is it doesn't fatigue easily. Okay, so whilst it has a medium speed of contraction, it does not fatigue as easily as skeletal muscle. So having a closer look at the anatomy of the cardiac muscle, okay, we're not going to spend too long on it. We'll notice again that we're just focusing on the intercalated disc, and that's essentially how it has the ability to pass on the stimulus to the rest of the cells. When you break it down microscopically, you'll see that there's a lot of similarities between the cardiac muscle and the actual skeletal muscle itself. Okay, so smooth muscles are very unique. Okay, they're nothing like the other ones. Okay, they're fusiform in shapes, means they're fat in the middle and they have long strings on either side. We have one nucleus per cell, which means they're more like your normal everyday cell. Okay, they're not striated. They are controlled involuntary, and the way they contract is more like a wave-like contraction. Okay, a very slow, methodical wave-like contraction, and that's obviously what assists us with our peristaltic movements. Okay, now the unique thing that a lot of people don't understand about smooth muscles is they actually have some pacemaker cells within themselves, so they can contract without the need of stimulus itself. Okay, now the stimulus to smooth muscles obviously comes by the nervous system. Okay, but we can also have hormonal stimulus that causes these muscles to contract. Okay, we think things like gastrin. Okay, so that's your smooth muscle control. When we look at the different types of smooth muscles, we have what's known as a single unit and a multi-unit of smooth muscles. So the multi-unit of smooth muscle has the autonomic nervous system just passing through it. Okay, so the stimulus can pass messages on to the cellular structures surrounding it. With a single cell unit, they have a specialized connection called a gap junction, which means the neural impulse will start at the top and the cells themselves will pass on that action potential through the gap junction. Okay, which is slightly unique and a little bit cool to do with the actual smooth muscles. In that way, the single units are similar to the cardiac side of the house. Okay, except cardiac obviously has intercalated discs, which means it can pass it more on a ripple effect all around it, whereas the gap junction actually means that as the membrane potential is charged, the action potential can be passed linearly down, if you will, from the autonomic stimulus itself. So similar in structure, but still quite different in the way it can pass messages on. Okay, skeletal muscles. Got to the point. This is where we're going to stay for the rest of the actual presentation. So skeletal muscles are long cylindrical cells. Now, these cells are very long. Okay, so a single skeletal muscle cell can be up to 30 centimeters long. Within that one cell, we have many nuclei. Okay, now the nucleus are there obviously to aid in your skeletal muscle repair. Okay, because remember, it's the nucleus that contains the actual DNA, okay, that can actually pass on the recipe for building muscle fibers to the ribosomes in order to construct the polypeptide chain. So that's why we have multiple nuclei all along the actual skeletal muscle, because if you only had one for a 30 centimeter cell, then it's not going to be very useful at all. Other cool thing about skeletal muscles is they're striated. Okay, and we'll find out soon where that striated effect comes from, okay, and what actually makes it look like a striation. They're under complete voluntary control. Okay, whilst you may do things that you believe is involuntary, okay, it is definitely your brain telling your muscles to do it. Okay? They can go through rapid contraction, but as a result, they can actually fatigue very, very easily. Okay, so they're extremely large, compact cells. They can contract with a huge amount of force, but they fatigue very, very easily. Okay, so that's what you have to think about when we look at the skeletal muscles itself. Now, when we look at the microscopic anatomy of a skeletal muscle, okay, we're going to basically start with the inner circles, if you will. So obviously, I know right back when you learnt muscles, you learnt the whole belly, and then you saw that it was broken up into its own capsules. Well, now we're grabbing one of those capsules and we're looking at that particular structure itself. So the outside, just think of it as any other type of cellular structure. We have a membrane, a plasma membrane, all the way around, encasing all of these muscle fibers. Okay, and that plasma membrane is called the sarcolemma. Okay, so sarcolemma means the membrane of the muscle. So a good thing about muscle words is it starts with a my, a myo, or a sa. You know that we're talking about something that involves muscle tissue. So the sarcolemma is a membrane that surrounds the belly. The sarcoplasm is just a very fancy way of saying cytoplasm for the muscles. Okay, so that's what that is. Now within the actual sarcolemma, we have a huge series of myofilaments. Okay, so these myofilaments, they stacked on top of one another, and they're long cables, basically. Each myofilament is a very long cable, and that's what makes up the cell itself. Now, surrounding the myofilaments, before we get into the actual myofilament itself, is specialized structures that assist in the overall function of the muscle. So the first thing is we have our terminal clusters and our T-tubules. Now, this specialized tissue is an extension of the nervous system itself. So it has the ability to pass on neural stimulus, continue, if you will, that action potential along. Encasing, like a big net, every myofibril is a sarcoplasmic reticulum. Okay, now I'm not going to dwell too much on what that does, but essentially the sarcoplasmic reticulum is vital in muscle contraction. Okay, so surrounding every myofibril is this sarcoplasmic reticulum. It goes all the way around, okay, not just what you see in the picture, but all the way. Now then, placed in between where it fits, okay, because the myofibril will actually take up about 80% of the entire structure, but shoved in where it can, will obviously have a lot of mitochondria, okay, because you'll learn soon that the muscle contraction and muscle function takes a lot of ATP, which means that we need mitochondria very close by, so then we can actually generate the energy that we need. Okay. Now, if we were to get a myofibril and pull it out, we'll see that it's made up of a huge amount of specialized protein chains. Now, these protein chains are what's responsible for contracting. Now, in between each section, basically, you'll learn to be called Z lines. So from Z line to Z line is what's known as a sarcomere. Okay, now we'll take a little bit closer look at that sarcomere, but just remember that when we're looking at the microscopic anatomy, this whole structure that you see before you is considered a myocyte. Okay, so an actual muscle cell. So the sarcomere itself is not the cell, but everything within the sarcolemere, sarcoplasm, and all the structures that we talked about before as well, that's considered the actual cell itself. Okay, so that's why I said that the cells can be enormous, is because, well, they can. Not like the normal cell that you would have been thinking about. So as we continue on and we look at that sarcomere itself, okay, we get a closer look. It's made up of two specialized proteins called actin and myosin. Okay, so the actin side of the house is thin. Okay, they're very thin proteins, and the myosin is what's known as your thick proteins. Now, your actin is made up of three specialized structures. Okay, so you have your G-actin molecules, you have your tropomyosin, which is essentially, think of it as a rope that ties those G-actin molecules together, and then you have a troponin that sits on top, something like a binding agent. Okay, so just remember those structures because they'll come into play mostly in the second round. The myosin, the thick myofilaments, have really heavy chains all wrapped together like cable, and then they have myosin heads. Now, it's this myosin head that is responsible for actually but facilitating the contraction. Okay, so that's the breakdown of actin and myosin. Now, when we look at their structure, we can see what causes this striation. Okay, so the striation effect of the actual muscle that we look at, the sarcomere, and how it's structured. So, breaking it up, the way that they've done it in a lot of textbooks is they've broken up different areas of control. Okay, so different structures that mean that they can easily define it without talking about large protein names. They can just call it things like bands and lines. So starting right on the end, we have what's known as the Z line. Now, in between each sarcomere, as they all bank up together, you'll have this specialized protein chain called the Z line. Now, the Z line is where the titans will actually connect to. Now, the titans will connect to the edges of the myosin and hold it at base. So one end will connect to a myosin, the other end will attach to a Z line. Okay, now these titans are coiled springs. So as the muscle shrinks and contracts on itself, it's the titans that allow it to go back to its original position. From there we have the eye band. Now the eye band contains just actin myofilaments. Okay, so it spreads over two halves, if you will, of two sarcomeres, and it just contains the actin filaments itself. The A band actually represents the length of the myosin. Okay, so the whole length of the myosin is represented by the A band. Now, obviously, part of that has overlap with actin and myosin, but the A band is supposed to be there representing the size of the myosin. Now, the M line is another specialized structure of protein that keeps the myosin where it's supposed to be. So the myosin does not move at all, as in the whole thing doesn't shift throughout the cell. Okay, it gets locked in place with the M line. Okay, M line being specialized protein that keeps it stationary. Now there is another one if you want to talk about the area that just represents the myosin. Okay, and that's called the H zone. Okay, the H zone in the middle, that just has myosin, in case anyone wants to be complete sticklers about it. Now, simply what happens in this sliding filament model, or sliding filament theory, is the myosin stays exactly where it is, and as you contract, the actin is pulled from the outsides in. Now, this is happening with every sarcomere, and the actin side of the house is pulled in towards the M line, and then at the point of full contraction, the actin will actually overlap itself over the M line. So the H zone will completely disappear. The Z lines will move closer together because the titans are actually shrinking in. Okay, and everything in the actin, the myosin separation will be gone. So the I bands will essentially just become the Z lines, and everything will push in towards themselves. Now in the next lesson we're going to go through the detail, the micro structures of how this works, but for now this is what happens. Okay, the actin's pulled in on itself, and that's what forms the contraction. Once the contraction is stopped, then with the assistance of the titans, it moves back out into its original resting place, the sarcomere at rest. Okay, and that's what happens all the time. We contract every time we move our fingers, every time we move our muscles. This is happening. Okay, so what controls this movement? What controls the need for this skeletal contraction is what's known as a motor nerve, and then subsequently motor units. Okay, so the motor nerve will come from your spinal nerves. So it's all controlled by your spinal nerves, and it'll branch out, and one motor nerve will branch out from the spine, and then it connect to your muscle fibers. So your muscle fibers, obviously we know they're all bundled up in a ball. So the motor nerve, once it comes in, will branch out into several motor units. Now each one of those motor units will essentially take responsibility for a number of different muscle fibers itself. Okay, so the fibers being what's made up from the sarcomere, the myofibril. Each one of these motor units, will be responsible for those themselves. Now each motor nerve can have a branch off before we get down to the axon, and it can have a branch off of anywhere between 4 different branches to several hundred branches. Now the difference is if you have an area that needs very, very fine dexterity, okay, so very, very specific but controlled movements, then you're going to have less axonal junctions. Okay, so you're not going to have more branches off from that motor nerve because it's easier to control. If you have something that needs to generate a large amount of force, then you're going to have several hundred of these motor unit branches that connect to different fibers around. Okay, so your back, your leg muscles, that sort of thing will have a huge amount of motor unit branches in order to control it. Now, what's a unique thing that we sort of need to keep in mind is muscles. So one muscle can be responsible for a small contraction all the way up to a large contraction. Now the reason that is because the motor nerve, as it comes in to control that particular muscle, it has a series of motor units that branch off, and they're actually responsible for different aspects of the entire belly itself. So everything found within the sarcolemma, okay, all those muscle fibers, each motor unit is responsible for contracting a percentage of each. So that way when we have a stimulus, we don't have to activate all of them. Now the reason that's an advantage is when I pick up a pen off the desk and lift it up, I don't end up throwing the pen across the room, okay? Or when I pick up a cup, a glass, a wine glass, okay, I don't end up crushing it in my hand. Well, not all the time anyway, but I don't end up crushing it in my hand because I don't have that full contraction every time my muscles get stimulated. Now this works on a principle known as motor unit recruitment. Okay, now the way motor units are obviously activated is they need a certain amount of stimulus in order to reach action potential. Once the action potential is met, then some of the motor units will activate some of the fibers, okay, based on the resistance that it's placed under. If that same muscle group is placed under more resistance, then you are getting more stimulus. Okay, and the more stimulus they get, the more myofilaments are instigated by action potential. Okay, so whilst action potential is still an all or nothing system, okay, it hasn't changed, I'm not reinventing action potential, you have an all or nothing system based on one myofibril. But with the increase of stimulus coming in, you can activate an action potential for more of those myofibrils. Okay, so more sarcomere chains are contracting on top of themselves. Now obviously there's a maximum capacity. Okay, so once you reach that maximum capacity, it doesn't matter how much more stimulus you put onto it, all the muscle is already recruited. So you can only do so much. Okay, that's why when you're bench pressing or squatting like the picture here, if you do a simple squat for a warm-up, barely any fatigue. Okay, because you're only recruiting the same muscle structures, but only a few of those actual fibers. Okay, so it's not that you're going through a weaker contraction. Each myofibril is going through the same contraction. It's just a matter of magnitude of how many we're actually recruiting. Increase the weight, we start recruiting a lot more. Soon we started recruiting the entire belly of the muscle, and then that's its maximum potential. Okay, so that's what motor unit recruitment is all about. Okay, and that really assists us. One, in sort of managing fatigue, so you don't have to go in guns blazing, all of them contracting at once, burning up a lot of energy when it's not necessary. And it also allows us to grade our response of contraction. So we have a lot more control over how much strength and how much force we want to generate because of this motor unit recruitment. Okay, so looking at the muscle contraction as a overview, so sort of as a concept of how it works. Obviously, a muscle twitch, whilst we say muscles are twitching, so obviously lifting a lot of weight, you get that involuntary spasms, that sort of thing. Now that's a little bit different to what we're talking about here. This is if we're in a lab and we get one muscle fiber and we stimulate that one muscle fiber with a probe. If we do that, then obviously we have a time period where the stimulus goes in. It takes time for the stimulus, for the action potential to pass on before the contraction goes through. So the cool thing about muscle fibers is the motor unit usually sits in the middle of the fiber, and from there the stimulus and the action potential spreads out. Okay, sort of like getting a stick and burning it in the middle, the fire burns out to the ends. Well, that's exactly what happens with the action potential itself. So you have a lag time for it to build up its charge. Once it reaches that area of voltage, then you get the contraction. The contraction will peak, and then once there's no more stimulus coming in, because we only did it once, obviously that membrane potential will drop back to resting potential, and then relaxation phase will come in. And that's essentially what happens when we're looking at it in an absolute isolated muscle fiber, muscle twitch. Now when we're looking at it in a practical sense, okay, the body uses something known as wave summation. This is a gradient curve of increasing stimulus or repetitive stimulus that causes an increased contraction, okay, an increase in tension. So as we try to lift something, we find out we get fatigued, and all of a sudden it gets a little bit heavy for us, so we get a new stimulus, and then it builds up our tension and recruits more fibers, and that happens each time. So what happens is as we're going down, the muscle doesn't have the ability to completely relax between contractions, but we can have this increase effect of the tension. Okay, now that whole thing is called wave summation. Okay, now that's what the idea of it is. It's a means of repetitive stimulus, or an increase in stimulus is what's known as wave summation. So the different types of contractions that we can get is what's known as incomplete tetanus. Okay, now tetanus obviously is, a lot of people refer to it as the disorder where you get lockjaw caused by Clostridium, I believe, and that's essentially where it comes from. Okay, so that's why it's called tetanus, is because it's got to do with fused muscle contraction. Okay, so in incomplete tetanus, we get up to the point of maximum tension, but then we still have the ability for some of the calcium, which we'll learn about later, to be recruited. Now what that means is the muscle has the ability to relax a little bit, so it can be sustained for that little bit longer. Okay, that's what we're talking about when we talk about incomplete tetanus. It's not a full strength contraction. We've reached maximum tension, but then we're easing off a little bit. Okay, so this is things that we can comfortably lift ourselves. Okay, so when we comfortably lift, we don't have to go to full tension. It's called incomplete tetanus. When we go to complete tetanus, on the other hand, this is where our muscle fatigue, we can only maintain that for a certain amount of time because of fatigue. Okay, we're not allowing anything to recover. There's no recovery stage. We reach up to that maximum force or maximum tension phase, and then we hold it for a period of time until relaxation must occur. Okay, so this is when we try to do our one max lift. Okay, we get it up. As we're lifting it, it's increased resistance against us, so we start recruiting more fibers all the way up. Finally, we get to the point that we're at maximum contraction, and we're holding it. Eventually, as we all know, we will have to release in order to go down. Okay, so using this theory of complete tetanus, this is where this concept of time under tension when you're lifting weight comes from. Okay, so the idea is that you'll lift a weight and you'll hold it until the points that you go through slowly, and then you'll slowly control it down, not allowing yourself to relax too early, optimizing this complete tetanus linear curve, if you will. Okay, so at this stage, calcium's just free flowing. There's no reuptake at all. It just sits in muscle, and obviously you keep burning energy as long as you're holding that contraction to an extent. Okay, two different types of contraction that we can have is obviously the contraction that shortens the muscle. This is your classic sort of contraction when you're lifting weight. We go from a elongated muscle and then we contract. Isotonic contraction means that we peak tension and our muscle length gets shorter because all of those sliding filaments have come in together. Nice and easy to go through. That's what's called an isotonic contraction. Okay, something else the body can do, which is a little bit cool, is it can go through what's known as an isometric contraction. So this is where you're holding something, okay, you are physically under tension, but you're not shortening anything. Okay, so think about holding something heavy in your arm. Your bicep's not shortening, your tricep is, but it was already shortened. But the idea is that your muscles can still get ready, so they can still go through tension, they can still make what's known as cross bridges. The myosin just doesn't shrink, so it just elongates, so this is what it grows the muscle laterally, but it doesn't actually shorten the length. Okay, so you're still able to generate force as a result of an isometric contraction. It just doesn't go through the same process as your filament theory. It just hardens up and create those cross bridges, and you hold those cross bridges for as long as you need to. Something else the muscles can do is as you go along and you're training your muscles, your actual muscles themselves can increase their muscle tone. Now what's happening with an increase in muscle tone is your muscles are more tuned to go through this contraction phase. Okay, so the minimal amount of movement is needed to activate the muscles to get ready for contraction. And that is translated as, when you palpate a muscle and it's actually tight. Even though they're not exercising or using it, you can still feel the tone within that muscle. And now that means the person has actually been training, it means the person's been working out, and the muscles are ready for the contraction. Okay, so what the muscles do, obviously this whole process, it needs a lot of energy. Okay, now in the next lesson we'll go through just how much energy is needed for this particular process to go through. Okay, and I think you'll all be very, very surprised how much is needed. But energy sources we spoke about in the last lesson, so we're not going to labour too much on them. I'm just going to mention them in relation to the muscle. Okay, so the muscle has three sources of energy that it can utilise. It has creatine phosphate, it has anaerobic respiration, and it has aerobic respiration. Now it will, as you start exercising, it will break through each one of these depending on its need and depending on what is available to the body itself. So looking firstly at creatine phosphate. So creatine phosphate is the first energy system to be utilised when existing ATP is used. So we'll learn that the muscle has ATP in itself ready to be utilised. Now that burned up in about 6 seconds. Okay, so the existing ATP is burnt up in 6 seconds, which means we need to recruit different energy sources to come into play. First one to be used is creatine phosphate. Now at rest, your body will come in and it'll actually break some of your ATP off and it will store it with creatine. So a creatine molecule and ATP will become creatine phosphate and ADP. Now that ADP can nick off, hang around, it's not being utilised, okay, and can be stored in areas of the muscle. The muscle already has storage of ATP, it only can hold a certain amount, so it can hold creatine phosphate and ADP as well as a byproduct. Now when the body depletes itself of ATP, it calls upon creatine phosphate to dump one of its phosphate molecules and give it to ADP, therefore reversing the effects and becoming ATP and a creatine molecule. Now what assists in this whole process is an enzyme called creatine phosphokinase. Okay, now we all know that one because we test it in blood, creatine kinase. That's what leaks out of the blood, out of the muscles when there's damage to the tissue itself. Okay, so it's that enzyme that comes along, cuts off the phosphate from the creatine, attaches it to ADP, giving us ATP and a creatine ready to be refused with phosphate during the next recovery cycle. Now, creatine phosphate energy system, you've got about 15 seconds. Now, obviously these numbers vary depending on fitness levels and muscle mass and all that sort of things, but generally speaking, that's how long you have before your creatine phosphate stores are burnt up as well. Then, depending on the level of activity, we'll go into 2 cycles. Now, if we're using creatine phosphate, then we are using high energy, high explosive sort of activities where the whole use of oxygen isn't really available to us. So we're doing high intensity training. Creatine phosphate's burnt out, and at the same time we start going into anaerobic respiration that we've gone through. Now, we all know that this is just going through glycolysis. So within the sarcoplasm of the actual muscle, glycolysis occurs, where glucose or glycogen, whatever it is, broken down into glucose, and glucose becomes pyruvic acid, yielding us 2 ATP to be utilised within the muscle. As a result of that, because we can't use the pyruvic acid to continue on, because there's no oxygen, it gets turned into lactic acid, back into the actual bloodstream to go to the liver to become glucose again. And the cycle just keeps continuing. If you remember the Cori cycle that we spoke about last time. So this whole system here, anaerobic respiration, you're good. You can generate enough energy before acids build up too much in the body. Oxygen depletion, damage starts to occur in about 40 seconds. Okay, so that's how long you've got for true anaerobic respiration. By that stage, the body has to slow down. The body has to get more oxygen in. Once oxygen comes into the system, we can start using that pyruvic acid. We don't have to get rid of it and transfer it into lactic acid. We can utilize the aerobic respiration, which calls upon glucose, it calls upon fats, calls upon proteins, okay? All the things that we spoke about last time can occur. And as a result of the entire energy production, we get 34, sometimes 32, sometimes 28, sometimes 38. Remember, there's a lot of arguments about it. ATP that can be used for the support in muscle contraction. So obviously aerobic respiration, that can continue for hours. Can't continue indefinitely because soon you're going to run out of stores. Okay, but You can, whilst doing activity, aerobic activity, you can continue for hours, depending on a few other variables within the body, but generally, energy speaking, this is your long-term one, this is your recovery one as well. Okay, so there's your 3 different sources that you can utilize within the muscles. Obviously, muscle fatigue is a thing. Okay, muscle fatigue is what happens when we can still get stimulus, but the muscles can no longer perform. Okay, so we're lifting a huge amount of weights. We get to the point that we drop our weight right down from 25 kilo bicep curl down to 5 kilo. We give it the stimulus, but we still can't contract. Okay, because we're in muscle fatigue. Now, things that cause muscle fatigue, okay, there's a few numerous ones. One of them is the absolute that causes it all the time. Okay, so the first one is if we start losing metabolic reserve. So the calcium is no longer there. The potassium, sodium doesn't have time to swap back to reset its membrane potential, okay, which takes energy as well, by the way. Then obviously we're not going to be able to continue to go through the contraction process. If there's damage to the sarcolemma, damage to the sarcoplasmic reticulum, as you could imagine, we're causing this huge elongation of blood and everything coming into the muscle when we use them, which stretches fibers, which can damage things. All of a sudden, sarcolemma can't pass on a beautiful membrane potential, an action potential, and the sarcoplasmic reticulum can't release or reuptake a good amount of calcium, so we start to get fatigued. But the main cause is the depletion of oxygen and ATP stores. There's only so much that can be made. So with the depletion of oxygen, obviously we have the build-up of lactic acid, and it's the lactic acid in combination with the other 2 that causes this fatigue. As a result of this product, we actually get muscle exhaustion and pain. So the build-up of lactic acid causes the muscles to be sluggish, and get really exhausted, and that's translated as pain. That way, so we need to stop. So other things that can factor in when we're talking about fatigue is obviously we've spoken about what muscle fatigue is, okay? But obviously psychological fatigue is a thing. That's either people giving up because of the pain, because it's too hard, so they stop, and that's their fatigue, okay? So it's a psychological block, hitting a wall, if you will, and it says it's not going to happen, okay? We can also have fatigue caused right at the neuromuscular junction itself. Okay, and that's due to a lack of acetylcholine that can be passed through. There's only so much acetylcholine that can come through. It needs to be recycled, which means that if we have something blocking it, acetylcholine can't come through, we have no stimulus. Okay, and that's essentially other things that can cause fatigue. Recovery is always needed. Once you do a lot of physical activity, recovery is going to be needed. And if you're doing daily activity like riding, sitting, drinking, all that sort of stuff, we're using muscles, we're using energy, but we've got a lot of oxygen. So we don't really need to recover because we're recovering while we're doing things. If we go involved in high intensity training using our creatine, phosphate and anaerobic respiration, then we need the recovery phase. Okay, because we've got oxygen debt, which means we need to get more oxygen in, and that's why the first thing to change is heavy breathing. Okay, if you start activity after being stationary for a while and you walk up a slight gradient, you will start breathing heavy. The reason is because you need this recovery. You need additional oxygen to make additional ATP proper. We need to remove that lactic acid. All the lactic acid needs to be picked up and go to the liver to be converted back to pyruvic acid and glucose in order to be stored as glycogen reserves. Okay, and that, remember, is called the Cori cycle. That all needs to happen during recovery. Okay, so that is part 1 of the muscle physiology. Hopefully you got a good overview of what happens in the actual body during this particular phase. Now the next one will focus purely on the contraction itself. So get real deep down into what happens when we contract the body. So until next time, take care.